This video is on cancer biology with a specific focus on throat cancer that in this case is occurring in dragons. Cancer involves a mass of cells that have an unlimited potential to divide and or grow with no use to the usual body functions. Most cancers are not due to defects in one gene but normally an accumulation of mutations involving several genes. The throat cancer that causes death in these dragons is thought to involve three genes, X, Y, and Z. My pet dragon Splash, who is pictured here, unfortunately had throat cancer, and although the exact cause was unknown, it is likely all three genes were involved. Splash was found to have benign tumours that remain localized and do not invade the surrounding tissues. These have both alleles of X mutated. For this gene and the following genes, the mutated variant is purple and the normal is a respective color, X being yellow. Although genetic, cancer is rarely heritable, but mutations in X inherited from previous generations can contribute to an increased susceptibility. Therefore, it is possible that Splash inherited one mutated allele and another normal allele. Most likely for this allele to mutate, a carcinogen will be involved. Splash also had malignant tumors containing cells that can invade the surrounding tissue. These again have both alleles of X mutated, but also have one allele of Y mutated. It is clear as only one allele is mutated that this involves a dominant mutation. Proto-oncogenes only need one copy of the mutant to transform to an oncogene, and thus it is highly likely that gene Y is a proto-oncogene. Splash's sister has not been diagnosed with throat cancer, so in comparison, her proto-oncogenes function normally and promote cell division through the release of stimulating factors. However, in Splash's case, as a dominant mutation has occurred, the oncogene will now allow for uncontrolled cell division. In order for this cancer to progress to the final stages and become metastatic, in which secondary tumours can be established throughout, an additional mutation in Z is apparent. This, as heterozygous, defines Z as a proto-oncogene. This table shows the number of alleles mutated in the contributing genes for different tumour types. All three types involve both alleles of gene X mutated. This is associated with tumour suppressor genes, as when abnormal, both copies are disrupted, leading to a loss of cell cycle regulation. Homozygous wild-type tumor suppressor genes prevent uncontrolled growth, whereas abnormal ones have no inhibition and result in excessive cell proliferation. Overall, Splash's throat cancer, although other factors cannot be ruled out, can be deemed a result of these contributing genes. The progression shows this particular throat cancer follows the classic cancer model, as numerous genetic events need to occur to cause death.